Hi, I am Daphne Hiporto from BS Respiratory Therapy 2. Welcome to Biochemistry, Central Dogma of Molecular Biology. Before we move into the main topic, did you know that James Watson and Francis Crick were credited for discovering this central dogma? In September 1957, Watson and Crick worked together on studying the structure of DNA. They proposed that the information that was transmitted has to be read by manufacturing body within the cell which puts amino acid together in a specific sequence, ultimately synthesizing a protein. What is central dogma of molecular biology? In molecular biology, central dogma illustrates the flow of genetic information from DNA to RNA to protein. It defines a process in which the information DNA is converted into a functional protein. It suggested that the information present in DNA is essential to make up all proteins, and RNA acts as a messenger that carries information through the ribosomes. This is the structure of central dogma. DNA makes copy of itself by the process of replication. Post-replication in every cell, the code of DNA is cracked and transcribed into a messenger RNA. This messenger RNA is then read and the code is translated into protein. Let's talk about replication process that we all know that it is semi-conservative in nature. Replication. Before cell divides, DNA must be replicated to ensure that each new cell receives the correct number of chromosomes. DNA replication process in which DNA makes a copy of the cell during subdivision. Part 3. Why DNA replication is important? DNA replication is important for cell growth, repair, and reproduction in organisms. Without the copying of the DNA life, it would not continue as existing organism and would not be able to reproduce and replace themselves. Life is dependent on the information stored on the DNA. In short, without replication of the DNA, the information would not be passed on and life would cease to exist. There are three main steps in DNA replication. 1. Initiation. 2. Elongation. 3. Termination. These three steps will be tackled later. These are the example of DNA replication from Team Insider video. Initiation. Before DNA replication can begin, the double helix structure of the DNA molecule has to be unzipped. Enzyme helicase breaks the hydrogen bond. Enzyme helicase breaks the hydrogen bonds that hold the complementary bases of DNA together, adenine with thymine and cytosine with guanine. This separation creates a Y-shaped called the replication fork. The single-stranded DNA binding protein, or SSB protein, binds to each single-stranded DNA, preventing the separating strands to join again. The RNA primer, which is a short nucleic acid sequence synthesized by enzyme primase, attached to each ssDNA to provide a starting point for DNA replication. Elongation. One of the strands is oriented in the 3' to 5' direction, which is known as the leading strand. The other strand is oriented in the 5' to 3' direction, known as the lagging strand. As the strands are separated, DNA polymerase enzymes start synthesizing the complementary sequence from the RNA primer in each of the strands. The parental strands will act as a template for synthesizing the daughter strands. DNA is always synthesized in 5' to 3' direction only. Therefore, in the leading strand, the template 3' to 5' it is continuous, hence called continuous replication. while on the login strand, the template 5' to 3', it is discontinuous replication. Thus, they occur as fragments called Okazaki fragments. DNA polymerase can make a strand in a series of small chunks. It is called Okazaki fragments. Each fragment started an RNA primer. This video is a summary of the leading strand is replicated continuously in the 5' to 3' direction. The other strand, called lagging strand, is replicated discontinuously or in pieces to 3' to 5' direction. In DNA polymerase, the enzyme that synthesizes a new DNA strand using a template strand can only add nucleotides to the 3' end of a polynucleotide strand. Overall, DNA replication is semi-discontinuous. Elongation. After the formation of both the continuous and discontinuous strands, an enzyme called DNA polymerase I or exonuclease removes all RNA primers from the original strands. The gaps where the RNA primers had been then filled by complementary nucleotide, another enzyme called DNA polymerase II, or proofreads the newly formed strand. In order to make sure there are no errors after proofreading, the DNA ligase enzyme fills the gap between nucleotide fragments.
At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one.
Termination. A special type of DNA polymerase enzyme called telomerase catalyzes the synthesis of telomere sequences at the end of the DNA. Telomeres are regions of repetitive nucleotide sequences at each end of a chromatid, which protects the end of the chromosome from deterioration or from fusion with neighboring chromosomes. Finally, the parent strand and its complementary DNA strand coils into the familiar double helix shape. The resulting product of DNA replication is two DNA molecules consisting of one new and new old chain of nucleotides. The human genome, or complete set of genes present in a cell's nucleus, is comprised of 3 billion base pairs. Remarkably, it takes very little time for our cellular machinery to copy this very long genome. Every cell completes the entire process in just 1 hour. Now let's talk about transcription. Transcription. The main motive of transcription is to make a copy of RNA from DNA sequence. Only one strand of DNA, called the DNA template strand, used during the process of transcription. As mentioned, the genetic information flows from DNA to RNA. This process takes place in the process of transcription, which is the first step in gene expression. The RNA that formed is called the mRNA. The mRNA or transcript carries the information used to encode a protein, like DNA replication. Initiation. The RNA polymerase is the main enzyme involved in transcription. It uses single-strand DNA to synthesize a complementary RNA strand. In the transcription, the role of the RNA polymerase, as you can see in the picture, RNA polymerase is the main enzyme involved in transcription. It reads one of the DNA strands and adds complementary nucleotide bases to make an mRNA transcript. The mRNA will later be translated into protein. RNA polymerase attaches to the DNA molecule and moves along the DNA strand until it recognizes a promoter sequence. These are known as the transcription start sites. The DNA-dependent RNA polymerase then binds to the promoter, which will then initiate transcription. Elongation. Elongation is the stage when the RNA strand gets longer by the addition of the new nucleotides. Once RNA polymerase is in position at the promoter, the DNA double helix unwinds, making all the bases on each of the DNA strands to be exposed. RNA polymerase walks along one strand of the DNA, known as the antisense or template strand, or DNA strand with 3' to 5' direction. For each nucleotide in the template strand, RNA polymerase adds a matching RNA nucleotide to the 3' end of the mRNA. The mRNA is nearly identical to the non-template, or coding strand of DNA. However, RNA strands have the base uracil in place of thymine. Termination. The process of ending transcription is called termination. The transcribed mRNA is known as the pre-mRNA. It is processed further to convert it into mature mRNA. mRNA processing includes capping, polyadenylation, and splicing. In capping, a methylated guanine cap is added to protect the mRNA. It occurs at 5' end of mRNA transcript. This protects the mRNA from degradation. In polyadenylation, poly-A tail is added at the end of mRNA. The poly-A tail consists of multiple adenosine monospates which protects the mRNA to degrade. In splicing, the non-coding sequences called introns are removed by the spliceosome. The coding sequence called exons are joined together by ligation. Finally, after this process, mature mRNA is obtained at the end of the transcription. Now, let's talk about translation. If transcription is the first step in gene expression, now the second step is translation. Translation. In translation, the mRNA is decoded to build a protein. The instruction in mRNA is read by the ribosomes to produce a sequence of amino acid chains. However, unlike in DNA replication and transcription, translation is not controlled by complementarity, but by the genetic code. A genetic code contains the sequence of nucleotides in mRNA that determines the amino acid sequence of proteins. It has a complete set of relationship among amino acid and codons, which is often summarized in a table.
What amino acid will that tRNA carry? Time to use the codon chart. We'll use a rectangular one, and remember, it's the mRNA codon that we look at, not the tRNA anticodon. So notice on the left side of the chart, it is for the first base. The top is for the second base, and the right side is for the third base. We're looking at the codon AUG. We go in order. First base letter, A. We isolate this row here and only this row. Second base letter, U. We isolate this column here and only this column. We've narrowed it down to this area here where they intersect. Third base letter, G. Here. The codon AUG codes for the amino acid methionine. Fun fact, methionine is commonly the starting amino acid.
However, the amino acid cannot read this genetic code directly and they need an adapter molecule. This adapter molecule is called tRNA. tRNA connects mRNA codons to the amino acids they code. One end of tRNA has a sequence of 3 nucleotides called an anticodon, which can bind to specific mRNA codons. The other end of tRNA, called the attachment site, which carries an amino acid. Each tRNA has specific amino acid attached on it. There are 61 codons per amino acid and each of them is read to specify a certain 20 amino acid commonly found in proteins. There are 3 more codons that do not specify amino acids. These top codons, UAA, UAG, and UGA, tell the cell when a polypeptide is complete. Initiation. In order to start translation, the few key ingredients must come together. This includes a ribosome which comes in as large subunit and small subunit. First, the ATP and the methionine bind to the amino acid tRNA synthase. Then, the tRNA without any amino acid attached bind to the same enzyme. The charged methionine is then transferred to the tRNA. The tRNA carrying methionine is released by the enzyme and attached to the small ribosome unit. After they bind to the 5' end of the mRNA by recognizing the 5' methylated guanine cap, then they walk along the mRNA towards 3' direction, stopping when they reach the start codon. After recognizing the start codon, the large ribosomal subunits immediately join to complete the initiation complex. Initiator tRNA binds to start codon. Large ribosomal subunit joins to form initiation complex. The large ribosomal subunit has three binding sites for tRNA. 1. The amino acid site, which accepts the incoming tRNA. 2. The peptidyl site, wherein the amino acid is detached from the tRNA and linked with another amino acid from the incoming tRNA by forming a peptide bond. 3. Exit site, which holds the empty tRNA before it leaves the ribosomes. Elongation. The methionine carrying tRNA starts out in the middle slot of the ribosome, the P site. Next to it is the A site, which will be the landing site for the next complementary tRNA. This step transfers the methionine from the first tRNA onto the amino acid of the second tRNA in the A site. It now forms a dipeptide. Once the peptide bond is formed between the dipeptide, the ribosome moves towards the 3' direction of mRNA by exactly 1 codon. The translation of mRNA into a long polypeptide eventually stops in the process called termination. Termination happens when a stop codon in the mRNA, such as UAA, UAG, or UGA, enters the A site. Stop codons are recognized by proteins called release factors, which fit neatly into P site. Release factor messes with the enzyme that normally forms peptide bonds by adding water. This reaction separates the chain from the tRNA and the newly made protein is released. This polypeptide often needs some edits during and after translation. Amino acid may be chemically altered or removed. These sequences, often found close to the N or C terminus, can be thought of as the protein's train ticket to its final destination.